IVM.
Hello and welcome to Simblified. This is part 2 of our interview with Avraneel Paul, scientist and a microbiologist. Part 1 of this interview was last week, and if you haven't listened to that, you should, because it will give a sense of continuity, and there's lots of fun, interesting things in there, including his own journey as a microbiologist and what the kind of stuff that he does right now, littered with loads of little facts around biology and science and what it is that a PhD actually does and what a day in their life looks like. In this episode, Naren joins us and we speak a lot more about a whole lot of other topics, including CRISPR. What are the potential new technologies that could change not just microbiology and biology, but the world itself? So that's on the other side of this. So thank you for listening to Simplified, and here we go. Welcome back to Simplified, and we are still talking with Avraneel Paul about all things biology, biotechnology, and all kinds of other delightful deviations. And this time we are proud to announce we have 50% more hostage. Does hostage come from the word hostage?
Yeah, some of our listeners do feel like it is a hostage situation, but I don't think that's the word.
So here's welcoming Naren to the show. Naren, hi.
I got delayed to this podcast, so I missed the delightful first half. But yes, I'm happy to be here.
So, Naren,
a lot of traffic today in Bombay, yeah.
So since you came late, you have to pay the usual simplified penalty. So Avraneel is going to ask you some biology-related questions, and he's going to test you. So Avraneel, feel free to ask some biology-related questions to Naren. Just ask him something that won't get a sidey response. That's the only thing that...
Naren, which is the most ancient disease of the world? Which do you think? The oldest?
That is not like a trick question. No, you're not going to say like some...
No.
It's not. A lot of diseases I know evolved later because back in the day people used to die happily in their 20s or whatever, right? So they never used to. So you didn't have diseases like diabetes and all, I believe.
Yeah, it is tuberculosis.
Oh, wow.
Yeah. Can you guess how long it has been on Earth?
So, Indus Valley? So 7,000, 5,000 years ago?
150 million years back.
Wow.
Wow.
Yes.
These lazy biologists, I tell you, like this, still not able to find a cure for that.
No, the reason why, Chak was asking, why cancer is so difficult to cure, why TB is so difficult to cure is like a very different question itself. TB grows so slowly. TB from one cell to become two, it takes 24 hours. Whereas E. coli, like the general bacteria, it takes 20 minutes. So you can understand how difficult it is to do any kind of research in tuberculosis. That is one point. And the other point is
what do you call the other bacteria?
E. coli. Escherichia coli.
E. coli.
Oh, E. coli. Okay.
The gut bacteria.
E. coli is the standard experiment bacteria, right? So there's the guy you use for testing all your antibiotics.
No, it makes sense that gut bacteria is solved quicker than tuberculosis because GB, as we know, is slower than TB. I'm very sorry. Just wanted to make that. So I'm very sorry. It's faster.
But so basically, if you're studying tuberculosis, most of the time your code is compiling, as it were, right? Like you have to basically wait for something to happen.
One code is compiling, and there are 5 other codes, which 3 we are working on, and 1 we are trying to solve the error.
So you mentioned you mentioned when we were starting that TB is a fascinating disease, and our biologist's relationship with it and solving it all. That it's a fascinating story. You said you use the word story, so fascinate us with the story that is like how humans conquer TB. Or
Yes. So as I said, that TB is the most ancient disease that has been found in human history. It has the first TB that was like the oldest TB that was found was in a sarcophagus of a mummy, Egyptian mummy. Like in a bone of a mummy, it was found, which was very another interesting case, that how a bacteria which infects lung can eat away your bone. That was very recently discovered in 2023. I have not read the paper yet, but I was recently watching an interview of that person. So that was the first time TB was found. TB was not called TB though. It was there in ancient Greek, ancient Rome. Ancient Greek it was called Pithis. Then in Rome it was called Tebis. And even in old Hebrew biblical texts you find the mention of this. And then from this ancient time we come to the Middle Age in England, where TB became very notorious. And at that point, I think 1 out of 5 people who were getting infected by tuberculosis were dying. But people did not know about TB. They were just looking at a disease which were making them cough. So cough is not a very specific symptom for anything. It's a general symptom. So people were coughing. They were producing red and yellow, the flame. So then that at that point, there was a notion that if somebody has this disease, they come to a royal attendance, and the king or queen or the monarch, if they touches him or her, he gets cured. So that was a common notion back then. So that's why it was called the king's evil. The TB was called king's evil. But still now people did not know this. I'm talking this about 17th century. So people did not know that why TB occurs, what is the causative bacteria. People did not know even about bacteria. So after a point from, I think, 1800 to 1850, many people come to understand that this is a disease, this is a new disease apparently, which is being caused by something else. Then finally Robert Koch, the father of microbiology along with Louis Pasteur, he isolated the bacteria from a TB patient. He grew it in another culture, like another petri dish and then a liquid medium. Then he took those bacteria and infected that in a guinea pig and saw the same thing is happening. So this was the first time TB, that Mycobacterium tuberculosis, such an old bacteria, was first isolated, cultured, and reproduced the same disease in another animal. So TB was found in 1870, I think.
My dad was a doctor, right? So back in the day, I remember they used to call it, he and his colleagues, and TB was a socially, so it was a disease which get you socially ostracized, right? So if people knew you had TB, they will not marry, into your family and all of that.
Yes.
Yeah. So doctor to doctor, they used to call it Cox.
Cox.
Oh.
Yeah. So Cox disease.
CHOCS. That is the Robert.
COCH. Yeah. COCH. Yeah. Cox.
This is
Cox.
This is Robert Koch who has first that isolation that has been done. That is Cox.
That's so
So
interesting.
doctors of that generation, they would call it Cox.
Yes. Cox.
So they would say he has Cox or she has Cox.
So as opposed to a viral thing, how does a bacterial infection spread person to person? Or does
Viral infection, virus is basically somewhere between non-living and living particle. Like if it's outside of a host, a living cell, it's just a dead particle which has a DNA and protein. If it gets inside anybody, then it will start its own life cycle and then cause the disease. But bacterial diseases, bacterial living organisms which comes into our body and immediately they start doing the damage, which is not the case for TB, which I must say. TB has a very interesting perspective of how the disease spread itself. It's not very common.
Do you want to tell us? I mean, you have a spider disease already, so go ahead.
TBD.
Yeah. So tuberculosis spreads by coughing or speaking or even singing. If somebody with tuberculosis sneezes, they can produce many air droplets in the vicinity. And each air droplet, considering how small a bacteria of Mycobacterium tuberculosis is, which is around 3 to 5 micrometer, In each droplet, there could be 3 to 5 bacteria residing in. So if somebody is inhaling that, Mycobacterium tuberculosis, which is the causative bacteria, goes inside the lung. And the moment it gets inside the lung, if it starts doing any damage, our immune system, the main, like the road sweeper of, there are many participants in the immune system. One of them is called macrophage, white blood cell called macrophage. So they are just the road sweeper, general road sweeper. Whoever they see their foreign particle, their dangerous particle, they will just eat it and then they will chew them inside and kill those cells. So if they are doing any damage in our lung, the bacteria, the macrophage will come, they will eat it, and then they will try to destroy the bacteria. But TB being TB, I don't know how it's so intelligent. It will just stop doing anything and it will just sit still inside the bacteria.
Oh, it's like it plays like it hides still like the danger of the road cleaners are passed by. That's fascinating.
No, not when the road cleaners are passed by. When the road cleaner has already engulfed it. It is inside that road cleaner. Now, it's not doing anything. It has gone to a complete dormant state. It is not dividing. It's just simply sitting there. Now the road cleaner, which has engulfed the bacteria, is thinking, what have I engulfed? Like, I don't know why I am, what I am doing here. I have cleaned this road. Maybe everything is done. So now that TB, the bacteria can sit there idle for a week, for a month, even for years. Like 25% of the human population has some bacteria sitting inside them. Like this is the WHO report. 25% of Earth population. Yes. So they can sit
other
there.
75% have no culture.
Wondering who would be the first one to bring that up, but it's finally happened. Short glass, everybody.
Yes.
Sorry.
Poor Avraneel.
So yeah, what happens? So they sit still, and then after a point, let's say after 1 year, they decide that this is my time to shine, and now they will start divide from 1 to 2, 2 to 4, 4 to 8. Now the moment they start dividing, that road sweeper, the macrophage, will understand something is wrong inside. Some foreign particle has gone inside me. I need to kill them. Now they would call all the other road sweepers that come. I am going to burst open. I am killing whatever is inside me. Whatever is coming out, please you guys take care of it. See that it does not get sprayed all over the body. So all the other macrophages come and surrounds that particular part, that particular cell. It bursts open. Like out of 100, it kills 80. And 20 gets outside. Other road sweeper catches them. Again, same cycle. They will go inside and sit still. Now there are 20 more macrophages which has been infected with tuberculosis, and they do not know what we are doing here. So this continues, and the more it continues, you can understand cells are rupturing themselves. They are bursting themselves, and when these macrophages burst themselves, there are very few harmful chemicals which they are releasing in that local area. So that will make the infected lung, like a hollow point, like a caseous point. We call it granuloma. So if that injury spreads more and more, it after a point at a very late stage, it touches the blood artery, like the arterial veins which are going through our lungs, which is basically exchanging oxygen and carbon dioxide. Like when we are breathing in and out, the blood is taking the oxygen and carbon dioxide by hemoglobin and throwing it all over our body. So if that injury is spreading, then it touches those artery and veins, ruptures it, blood comes into the lung, people coughs and produces blood. So that's why TB coughs are often red in color. So this is how tuberculosis spread.
That was graphic and gruesome.
I have an interesting. I'd read this in a book on the Second World War. So during the end of the World War, Berlin, Germany actually especially Berlin, was attacked by both the Allies and on the other side, the Russians. And the Russians had they were just, what they would do was, they would rape women, basically. That's what they would do, and they were, it was a crazy time, right? So what women used to do was they would cut their finger when they saw, they would have a handkerchief. When the soldier came close, they would cough and show the handkerchief. And the Russian soldiers were terrified of TB. So they would see that and they would just run away. So that was the
Yes.
hack. It is very macabre, but I remember reading this in some book.
Yes, very possible.
Yeah.
Very much possible.
Have you, Avraneel, have you read Project Hail Mary by any chance?
I am planning to read. Like,
Yeah,
it's
you
in
should,
my-
because you kept saying macrophage, and in Project Hail Mary there is an interstellar microbe called an astrophage. So it just reminded me of it. I don't want to spoil it for you,
So
but it's a
basically
fascinating book.
out the asteroids, is it?
No, not asteroids. I suppose the astro comes from the sun, and I mean, I don't want to spoil it for you, but it doesn't eat anything as such. I think it's more about what it gives up. That's interesting.
We used to have a professor of electrical engineering named KJ Singh. So this KJ, very good professor actually, but unfortunately, he would pronounce phase as phage. So he would
PHA
keep
is
talking
phage.
about three-page circuit,
Three-page.
single-page circuit, he will say like that. And so if people would, engineering college guys being engineering college,
Oh.
they were like, somebody had come up with an idea for a KJ Singh restaurant. So
Oh,
KJ Singh
no.
restaurant, you would get veg, non-veg and three-page. So I just remembered KJ Singh. But wherever you are, Mr. Singh, you are one of the best. So, yeah.
But talking about the phage, do you know like what is bacteriophages?
Yeah.
Tell us.
Yeah.
I mean,
So
I think
Bacteria eats a
already.
bacteria.
Exactly. Not eats a bacteria, but kills a bacteria. So it's a virus that kills a bacteria. So this like research of bacteriophage has given many insights in many different fields. One of them is CRISPR. Like CRISPR, the gene editing tool, had nothing to do with gene editing. It was first found as the bacteria's defense mechanism against those bacteriophages. So that's another like different story we can talk about. So what bacteriophages do, it usually attaches to a bacteria, it injects its DNA inside, and then the DNA is like read by the bacterial system, like bacterial machinery, DNA machinery, and it produces viral protein. Now the viral protein will assemble around its own DNA and produce new viruses inside the bacteria. So this is how a bacteriophage infects bacteria. So scientists, when they were studying bacterial DNA, they saw this random repeats in the bacterial genome sequences, which were not of bacterial origin. So when they started studying, they understood this random DNA sequences in the bacteria is coming from the bacteriophages, the viruses. Now, how is that happening? It's a very beautiful machinery how bacteria develops. Like, for me, it's very fascinating because we talk about immunity from a human point of view, where there are millions of different cells that are fighting against different microbes, different pathogens. And here there is bacteria, which is a single cellular organism, is creating an immunity of its own. How is it creating that? Like every time a virus infects it, if it's able to kill that virus, it cuts a copy of the viral genome and put it in its own DNA. This keeps happening. Like every time a virus comes and a bacteriophage comes and attack the bacteria, it will keep a copy. Now this copy serves as a memory that if next point, if that same virus or the same family of virus attacks, I know that this is a foreign DNA. I have a copy of that. I will go and just chop off that DNA. It's a very like sequence specific DNA cleavage. Like it reads the DNA sequence and matches it with its own memory copy, go and cleaves it all up by a nuclease enzyme called Cas9. So that's why the term called CRISPR-Cas9.
CRISPR,
So this is how CRISPR was.
I think this might be a good time to talk. Just talk about CRISPR, something that I've been meaning to ask you ever since you kind of wrote in. But before that, since you spoke about this aspect of memory, I remember reading a long time back about how biology-based memory systems might actually be developed. Is this actually a thing, or did I just dream it up somewhere? Like what I mean is like from say from your regular spinning hard disk to solid state device to whatever quantum is up to right now to eventually biology and cell-based memory. Is that something that's actually happening?
Possible, but I have not read about it. But logically speaking, this will be even harder than producing a hard disk or like SSD. Like, it might be efficient, but would be very hard to make one.
Retrieval,
I have not read about it. Yes.
But
Yeah.
what fascinates me is this, right? This whole thing is chemical. So everything that's happening inside the bacterial cell, the DNA, the pieces of DNA that are taken out, snitched in, there are enzymes and facilitators that make this possible. They get stitched into particular locations. Copies are made, then get stitched. The whole thing is it's just chemistry.
Yes, chemistry.
And yet, so I believe CRISPR can replicate that. So you can replicate this chemically, right?
Yes.
So
yes. Yes.
So now the boundaries between is a question for later actually. So we'll come back to it. I'm really keen on understanding what CRISPR is all about. So I'll let you go on. But what fascinates me is
episode that we did on CRISPR about 2 years ago. We actually did
we just went right over.
We just went right over.
CRISPR is one of the things. You can have it explained to you 100 times, and I won't get it even on the 100th time. So I'm always going to
let's try for the 100 one time and the last time.
Yeah. Let's try.
So no, my point was where does chemistry end and consciousness begin, right? So if bacteria are chemical, so are we all.
Yes.
Right? So because we are really not that different from bacteria, and yet we have something called consciousness. So we have experiences which are like not explainable by just chemical things, at least not that we know of. Yeah. So let's come to that later. I just thought it crossed my mind.
Let's talk about CRISPR actually. Let's talk about CRISPR.
So CRISPR first.
Yeah. So a brief introduction to what it is and what's actually the state of things right now. And I would be very keen to see what role, if any, CRISPR played in the events of the last 2 years.
What is CRISPR? According to you, you have also heard about CRISPR. What is this CRISPR?
It's a system for gene editing. It's the simplest that I can possibly think of, which has drastically reduced the price for gene editing and gene-based research, and has the potential to solve a whole bunch of diseases. Yet there are ethical problems like designer babies and all that. This is sort of like what I remember from the research for that episode a long time ago and whatever I've been reading of late.
Yes, so absolutely correct. CRISPR is a gene editing tool. That's a basic. But what's so special about it? Gene editing tool is nothing new. So to understand that, I will tell you my story how CRISPR saved my life. So when I came to
that's a hook.
How CRISPR saved. No, not my life, not my real life, my research life. Yeah. How
also impressive. Not as catchy as the clickbait that you led with, but let's go with that.
Yes. So what happened when I joined this lab? I was working on a particular gene. Now, gene can be knocked out in any DNA, any organism, which is a very well-known fact. Now, the problem with this gene was, this was an essential gene. Essential gene means if you knock that down, the bacteria or the virus will not survive. Now, if the bacteria itself is not surviving, how can I study that? So that was the biggest issue I faced. So to bypass that, I had to make a very difficult system where I have to put some 2-3 plasmids in a single cell, then express the gene on my condition, then knock out the native gene from the bacterial body, which is a very, very long process. And mycobacteria being such a slow organism, you can understand any cloning is also that slow. So I was struggling with that in my lab when I came across a CRISPR paper which has used this CRISPR technology to silent a gene. Like there is a scientist in Rockefeller University called Jeremy M. Rock. Like he was trying to study the roles of different genes in mycobacteria. For that, he had developed a tool where he can produce a protein which will not cleave your gene, like which will not knock out your gene. The gene is still there, but at a particular condition, if you are giving a particular antibiotic, for example, if I am giving antibiotic X or chemical X, this system will get activated and it will just suppress that particular gene. So the gene is there. So it's not, I am not completely deleting that gene, but for a moment, when the X chemical is present, that gene is silenced. So for that moment, I can study that system. So this is called CRISPR-I or CRISPR interference, which is a modification of the classical CRISPR system. Now, what is classical CRISPR system? It's very easy. It does not take you months or years to develop. You just design a plasmid. Plasmid is basically a round structure of DNA, which you can insert in any cell, like by electric shock, by chemically permeabilizing the cell, you can put that inside. So you just put inside a plasmid, which has a specific sequence. Now you remember when I talked about the virus memory, I told you that they were very much dependent on those foreign sequences. Yeah. So you want to, if you want to suppress or delete edit a particular gene in your whole genome, you give a specific sequence, which is just 20 AGTC, those DNA bases added in guanine, thymine. So 20 different permutation combination of this AGTC, you attach a Cas9 a protein to it and you send that thing inside a cell. It will automatically go detect that particular sequence and it will suppress it or edit it or delete it or activate it or make it visualize. So this is what makes CRISPR so easy to use. You just have to give a sequence, a 20 AGTC sequence, and you send that plasmid inside the cell and tell the cell you do whatever you want. So that's CRISPR in a simple language.
You said it's easy to use. So easy for whom to use and how? Like, can I use CRISPR over here sitting in not sitting at home
if you have a method by which you can take those plasmid and insert into any cell, yes, you can. Theoretically, so you can.
Yeah, theoretically. So you need some level of the infrastructure to make it happen.
Yes,
right? It's not you can create. Yeah. This is sort of CRISPR was as it is fascinating / scary before this whole generative AI thing just exploded. And this is, I'm sure, something that you have been thinking of, and your community has been thinking of as well. Like, is there a intersection between the two? Is there an intersection between, say, large language models and technology like CRISPR? Like the simplification of I could ask, is there a crossover between ChatGPT and CRISPR? But I think the question is larger than that. The role of AI in biotechnology overall. That's basically what I'm trying to get at.
AI is, yes, machine learning or the language system is very fascinating and it's currently seeping in the biology field as well. But AI is mostly used for literature purpose, right? Like you ask it to generate, you give it a prompt and it generates you a certain text. So it's just text based. So maximum I can understand, if like best case scenario, you can ask a chat GPT that can you give me the sequence to silence gene X. If it
Yeah, that's
knows
what I
the
meant actually.
sequence, yeah. But that finding the sequence manually also does not take that much time. Like we have softwares already present, which is not AI based. You can give a prompt that I want to like knock out or knock down this particular the gene ABC. So they will give you a 20 sequence. You can use that and knock it out. So it's not needed right now.
It's not,
But
it's not needed, I suppose, for scientists in the field, but I can see how this, how some of the more, say, doomsday scenarios with respect to CRISPR can actually be more democratized with tools like this, where you just need to literally enter a prompt and there could be an output. But maybe that's a discussion.
I have one. I have one more question. If you can change genetic code using CRISPR, how do you, if I want to, let's say I have some disease, which is genetic, and I would like to knock it out. How do I ensure that the genetically modified thing reaches all the relevant cells?
Yes.
How is that? How is the delivery done?
Question.
Yeah,
that's a beautiful question. So when you say that, like, I thought the question was going to stop at how I'm going to cure it, but it went further. So the thing is, let's say somebody has a sickle cell anemia, which is a genetic disorder. Now, sickle cell anemia means they have a problem in all the red blood cells, right? The beauty of our body is that we were all a cell, a single cell, then a bunch of cell, and from that we develop, right? So you can understand there are always that those original cells, like if we call it stem cells. So original cells are there. Like if I'm calling like red blood cells, there is a generator of that red blood cells. If I'm calling, let's say, the recent advancement in this CRISPR technology, a girl in London had a T cell leukemia. So her T lymphocytes was corrupted. So there is a cell or a particular type of cell which generates this lymphocyte. So scientists have engineered that cell and put that inside her body. So now as the origin or the generator itself has been like modified, now they are producing the modified cells. So that's how you edit from a basic point, not to the last end node from the beginning.
Okay, so the source is what you edit.
Yes.
Whatever is existing. So you're basically trying to modify the new cells which are coming up. Whatever is there in your system will eventually die or just senescence or whatever it will happen. Yeah. Okay.
Absolutely.
So that's why I guess you can't do anything for brain cells, because I believe brain cells are not. Yeah.
It's very
damn. I wanted to be smarter. I thought I'd take a pill and no.
You are. You are. As we're
yeah. Actually, this conversation is already making me smarter. So yeah.
So, no, fascinating. So we've asked you our opinions of a lot of technologies, AI, CRISPR, everything. But what are the sort of like technologies or even like trends that excite you? And you asked both Tony and me what you think I do on a daily basis, but what is it that kind of motivates you? What are the kind of technologies and things that you hope to see come to fruition in some sense in about 5-10 years and sometime over the course of your career? Does that make sense?
One would be to eradicate this like age-old disease. If we find any cure which can cure it faster, of course, because MTV's tuberculosis treatment is very, very like painful. Like you have to take a regimen of like a 6-month regimen of different sets of antibiotic. So if there is any miraculous cure for this, would be great. Other than that, there are a few trends that, of course, CRISPR, like thinking to edit something so easily itself is very fascinating. Like as I have told you, to edit or to delete something takes a lot of time. And if you can if like you have a tool which can do it like this and anything, and CRISPR could be like scaled up for a like at a large extent. From one gene you can delete 5 genes at a time. You can delete 5 genes, suppress 3 genes, activate 2 genes at the same time. So you can use it in all permutation combination. So this is something. Then cancer immunotherapy is one more thing which is very fascinating for me, where you are training your own cells to go and kill your own cancerous cells. This is a very interesting topic. And then other than that, like biomimics are there, which are like looking at the nature and then engineering those same things in your laboratory and produce. For example, geckos, like the one which can walk on water, can hold itself on a ceiling, the chipkalis. So like the chemicals that extracted from this, like the gecko paw, that has been re-engineered and made those gloves you have seen in Badshah, like the Hindi movie, people like walking Spider-Man type of, that has been engineered. Those super glues has been engineered. Color has been extracted. Synthetic dyes has been extracted from different colors from butterfly wings. So these are the things which has been in development for quite long time, and now with synthetic biology, computer codes coming in our field, it has been revolutionized. Things have become very faster, I would say.
One more thing, does this worry you that so there's a lot of buzz about the COVID virus being an engineered product, right? Mm-hmm. So, and the jury is still out. So some people say that yes, some people say no, it was, whether it was lab created or
yes. So,
but you do have the platform for bad actors or for whatever reason to do this kind of thing. So does that worry you a lot, or do you think that it's something that's that the scientific community will be able to handle if and when it happens?
I can understand why that debate is still there, that it's a lab produced virus, lab engineered virus or not, because it's possible. Like if you have a virus or bacteria, and with CRISPR or any genome editing tool, you can just engineer it anyhow you want. So we understand how it is done, so it's possible. But again, from an ethical point of view, it's very hard to believe for us that somebody will create something. I know it's possible, but dil manta nahi hai. That's the problem. So it can be.
I have another question. So these, they, for all I know, they probably already used. But do you see that as a possibility for greatly ending world hunger or whatever that it may? Because you could really engineer plants, and the people are doing this lab-grown meat also. So is that? Do you think it's likely in our lifetimes? In my lifetime, wow, you guys are much younger, to see something like this, a post-plenty kind of thing, where really nobody needs to go work at all because salah apne aap ukta hai sab kuch.
Yes. Hopefully, yes. Because those genome modified plants, the genetically modified plants have been in the market for a long time. Good for tomatoes, then self-protecting maize. These things this were already there. Now the lab-grown meat has come to the scenario which has advanced this food industry from fathom. But to cure world hunger, I think we need to get this technology and make this reproducible in the remotest part of the world. You or me sitting in Birmingham or you sitting in Mumbai is not facing that much of scarcity of food, but a kid in let's say anywhere in the African country, let's say. So they are suffering from that hunger. So taking this meat and giving it to them is the bigger deal right now, because I think we are producing a lot.
We really don't have a scarcity. Yeah, it's not a production problem; it's a distribution problem.
Yes. And the other thing is, the more this technology is advancing, we are hoping that it will be more cheaper to attain. So that's a hope.
I have, I mean, we've asked you, we have pretty much grilled you about, your
I'm
career.
having
And
a great fun.
I have sort of like one last question. And it's actually going back to something that you mentioned at the earlier first half of this conversation, which is that, most scientists don't like talking about themselves and their work, or at least that's sort of like the culture. Do you think that that's something that needs to change? Because the way I look at it, it feels like we need slightly more vocal voices in the space of science. One, from a policy point of view. Two, from maybe just energizing the masses and, getting them excited about science in the way that people from Carl Sagan to, say, even a Randall Munro have in their own different sort of ways. And I know that there are some scientists who are pretty popular on Twitter, etc. as well. I remember the case last year of somebody who had actually done a very weird kind of research paper, and it was very meta. I think it was about changing the structure in which most research is the way in which research is written in your standard papers. They're very dense and they're very boring, and because of that, people aren't getting more interested in science. And he used his paper as a meta means of communicating exactly this point, which I thought was absolute genius, and that had gone viral beyond the PhD community. So the point I'm asking is, do you think scientists should actually invest a little bit in, say, PR for themselves, maybe even make it part of courses when they are studying? Because I do think that's something that's going to be slightly important going forward. So I'd like to know what you think about that and what you see, what you kind of observed this. Do you feel like this is a need as well?
What you said is very, very true. Because if I just take up any random paper and I just start to read the abstract, it's very heavy. It's very jargon heavy. But at the same time, this is not like the common or the undergraduate is not reading all the research articles. The research articles are the articles which are getting published in Nature, Science, PNAS, etc., etc. Those are hardcore scientific studies. But the good thing is, right now, the scientific community has been able to trickle down those bunch of heavy knowledges and made it easy to digest small tidbits. Right now, even TikToks, YouTube Reels or Instagram Reels have come where people are summarizing a whole such a difficult paper within 1 minute, just with the important points. But yes, we are not a very vocal community. We are very vocal within ourselves. We will go to 5 different universities, give lecture, and the undergrads and masters will listen to it. But we are not that vocal yet. I am sure that people are coming to this field. Right now, scientific journalism is a big thing, where people are taking these knowledges and simplifying it for larger audiences. So I'm pretty hopeful that it's happening right now. So,
I have one phenomena conk level question. Is it true that, is it true? Now you have to give an honest answer, but is it true that the only reason people want to become molecular biologists are because they get to wear designer jeans? Is that the reason? I couldn't resist. I think
the only reason people want to be molecular biologist is because it sounds nice to say that I'm a molecular biologist.
Hey, what's the difference between a microbiologist and a molecular biologist?
So microbiologist is the one who works in microbes. That could be, for example, I am a molecular biologist, I'm a biochemist, but I work in microbiology. So molecular biologist is if you are doing anything with the molecules of that. For example, I'm working on a protein, so I'm a molecular biologist. So anything with the molecule is a molecular biologist.
But because you're working with microbes, you're also a microbiologist.
Microbiologist.
Okay.
If I was working cancer molecules, I would be an oncologist or like cancer biologist, not oncologist.
Reminds me of a friend we had in MBA. His name was M. Balaji, and the way he would sign his M looked like a mu. So we used to call him microbiology.
About speaking of micro, the mu, the symbol mu, it's also the symbol for the coefficient of friction. You know that, right?
Yeah.
So the question is,
It's working
if you
its
have,
way towards a cat joke.
That's why the cat is
everyone knows
silent.
that. Oh, man.
Narayan.
Oh God.
Narayan. Have you forgotten?
Fell flat.
Why does a silent cat slide down the roof?
No, I think we just for the like listeners or viewers, if you want to solve questions, like if solving any mathematical problem or any problem interests you, then just come to this field. You will get a tons of questions to solve, and that will take you 5 to 6 years, of course. But you will ultimately solve it.
For somebody who doesn't want to invest, say, 5 to 6 years and just wants to sort of like, I'm going to call this pop biology. Like, are there any YouTube channels, any podcasts, anything that say does a better job of what the four hosts of this show can do, simplifying things, and maybe like sort of like what say a Randall Munro does for physics or something like that? Any resources that you can recommend? Any books or anything like that?
Yeah. First, I will come to podcast recommendation. So there is a podcast called Sci-Fri, Science Friday, and which is a great podcast, scientific podcast for general people where they do not fire jargons to you. And then for microbiology, there is one more podcast from ASN. It's called This Week in Microbiology or This Week in Virology, which is a great podcast where they discuss the current publication of the last week. And those are very interesting. Recently, they have been talking about a study where it's called where the replicas does not replicate. Where they have found a culture of bacteria which has been generated from the same bacteria. So they are supposed to behave identically, but they are behaving in 5 different manners. So that's a podcast recommendation. And books, I have recently been reading a book by Siddharth Mukherjee, which is called The Song of Cell. So that's a very fascinating book where he has talked about cell biology, what a cell does, in a very storytelling manner and anecdotal way. So it's my recommendation.
Those are some recommendations. Thank you for those. And honestly, thank you for coming onto the show and sharing your experience, wisdom, and the stuff that you're doing. And we know that you have a few plans, dreams around it. So can only wish you all the best for those. I think the planet will be better off with folks like you undertaking these hard problems rather than us just blabbering into a mic like this once in a while. So yeah, thank you for reaching out as well. And this would have happened if you hadn't pinged in. So yeah,
this was awesome.
Thank you.
But coming back here, so Naren, the usual way to sign these things off.
So thank you very much, Avraneel. So it was a great episode, and stay safe and stay CRISPR and stay simplified. Or stay molecular and stay simplified. Yeah.